All right, hello class. We are back at it. This is the second lecture in the course. This is the second lecture in the chapter on the special sense organs. Last time we met, we were talking about the eye. We had learned about the special sense organs for the sense of smell. We learned about the anatomy and physiology of gustation, a sense of taste. And we had moved into the eye, and we were covering the accessory structures of the eyeball, all those structures that are associated with the eye, about maintaining the health of the eyelids, the tear glands and ducts, and we were just finishing off this section on the accessory structures of the eye. Okay, so tears. Okay, we have to know where they're produced, how they flow from their source into the eye, and then where they drain. That's what we had talked about. The puncta are the openings that are medial. The canaliculi are the ducts that are taking the tears from the eye into the lacrimal sac. Lacrimal sac leads to the nasolacrimal duct, which connects the eye to the nose. So that wraps up the accessory structures. Now let's deep dive into the eyeball itself. Okay? This is a sagittal section through the eye. We can see lots of structures here. We're going to focus here on the edge of the eye here, because the eye is a hollow structure. It's filled with fluid, and there's 3 layers here that we need to know. Okay, 3 layers to the eyeball. Okay, starts on the outside, and there's a middle layer, then there's an inner layer. They have 3 different terms. The outer layer is called the fibrous layer, or the fibrous tunic. It's comprised of the sclera and the cornea. The middle layer is called the choroid, or the one with the blood supply, the vascular layer. Sorry, brain fart. Okay, the choroid, or the vascular layer, vascular tunic. And then the inner layer would be the neural layer, okay, which is the retina. Okay, so we're going to go through each of those layers in sequence throughout the rest of the slides and talk about each of these layers. Okay, around the tops and sides and back of the eyeball, those layers are very easy to distinguish. As we get to the front here, things get a little bit messy, but those layers are still in place because the fibrous tunic, or the outermost layer, becomes the cornea. So we'll talk about the cornea. Then we'll talk about the vascular layer, or the choroid layer, which becomes part of the iris and the structures that are supporting the lens, okay, which we'll talk about. The retinal, or the innermost layer, the neural layer, only extends about 3/4 of the way towards the front. Okay, from about here to here, there is no neural layer. Okay, and that kind of makes sense because the neural layer is the retina. The retina is receiving the photons of light. So light is starting out here and is entering into the eye this way, and it would strike the retina and then interact with the rods and cones there. All right, so we are going to go through those structures in more detail in the coming slides. So here's the 3 layers: the fibrous tunic on the outside, the vascular tunic in the middle, and then the innermost in yellow on this slide is the retina. This gives rise to, we mentioned that the eye is hollow, okay? And so there's 2 cavities, okay? There's the posterior cavity and the anterior cavity, okay? And the division point there is the lens, okay? The lens defines the boundary between the posterior and anterior chambers, okay, or cavities. Okay, so the anterior cavity has something called aqueous humor, and the posterior cavity has vitreous humor. They are slightly different in their consistency. One is more of a gel, and one is more of a liquid. The anterior cavity has the aqueous humor. Aqua means water, so it's more of a liquidy consistency. Okay, so there's fluid that's flowing around inside and in front of the lens, and actually behind, a little bit behind the iris. It's shown in this kind of pink color. And then most of the eyeball is filled with the vitreous humor. The vitreous humor is more of a gel-like consistency, and it is continually providing pressure to keep the eyeball kind of inflated, as it were. Okay. Here is a superior view of the eye. Here's the nose. Okay, so we're looking down, kind of bird's eye view of kind of a quartered section here of the eyeball. And this gray dotted line is showing you the visual axis. The visual axis just means how does light enter the eye? Okay? All vision starts with a light source. Okay? Right now in this room we have fluorescent bulbs, okay, that are, electricity is coursing through them, exciting the fluorescent bulbs, and it's emitting energy in the form of photons. Those photons are flooding down into the room, scattering every which way through the diffuser, and they're bouncing off of objects. Okay, like right here, I have a purple marker. The light is bouncing off of the purple marker, and then some of that light is scattering and entering into my eye. Okay? So the light starts there, bounces off at a very specific wavelength, and then that refraction enters my eye, and I can see the marker because of the bouncing light. Okay? That's important. Okay? So all vision starts with a light source. If it's dark, we can't see anything. Okay? If we were to turn the lights off, there's no vision because there's no light source. It's a bouncing light that enters into our eyes. So this is the entering of those photons of light. A photon is a discrete packet of energy, okay, that will enter through the cornea, through the lens, through the vitreous humor, and hit the retina. Okay? It's the retina that receives that photon and can distinguish different wavelengths of light. Right? So there's lots of terms here that we will go through in turn. Okay? But this is a beautiful picture. You should be able to identify all the terms in your notes outline and be able to identify each of these structures. The outermost layer here, the sclera, that's the fibrous tunic. As it approaches the cornea, it goes from a white color to a clear color. There's no color. Okay, so a clear appearance. And what you need to know is that they're both made of the same material. They're both made up of a lot of collagen. It is merely the arrangement of that collagen which makes it transparent, okay, or clear. So the cornea is crystal clear. It allows light to pass right through without any or without much variation. Okay, the sclera does not allow light to pass through, okay, because its arrangement of collagen is different. Okay, so here's the cornea, which is the clear part. It allows us to see through and see our iris. The iris is the colored portion. You either have brown or green or blue or some variation of those colors. The iris is the colored portion. And then the hole there is called the pupil. Okay? And the hole provides access to the lens. The lens also is a disc-like structure that is mostly collagen, and it is arranged in a disc of connective tissue that is very flexible, and it's quite elastic. So that as we have attachments, think about a round trampoline, okay? And around the edge of your round trampoline, you have these springs that are kind of pulling the trampoline tight. If you were to pull it really tight, the trampoline would kind of flatten out. But if you were to relax the springs, well then maybe the trampoline could then, if you imagine this as a disc instead of a sheet of whatever that black stuff is, okay? The lens can be stretched thin or allowed to kind of plump up and get thick. And the thickness or thinness of the lens can change, it's called accommodation, to focus the light from different sources. Something that's really close to your face, like you're trying to get a sliver out of your finger, okay? The lens will change shape, rather than if you're focusing on some distant object like a traffic light that has just turned yellow and then red, okay? The lens will change shape to account for different focal lengths. and allow for the distant object or the near object to be correctly focused onto the surface of the retina. Controlling the iris and controlling the lens are muscles, okay? They're smooth muscles that are controlling the diameter of the pupil as well as the thickness of the lens, and those are controlled via autonomic feedback loops. Your rectus muscles here can attach to the eye and help position the eye to look side to side, up and down. Although our head does a lot of moving when we're scanning our visual field, our eyeballs can also scan as well. This bluish color here is near the front is called the ora serrata. It is really the interface between the retina, which is ending, and merging with the ciliary processes. Okay? The ciliary body and the ciliary processes are these muscles that are controlling the lens and the iris. They're circular, so think about them as more like a donut, okay, shape. And they're attaching to the iris and to the lens. All right, so we just talked about the fibrous layer. Two parts here, the sclera, which is the white part, and the cornea, which is the clear part. Okay, so all of it is fibrous layer. It just changes consistency when we get to the part where the light has to enter. The limbus is just the border between the two. Okay, the vascular layer has multiple functions that you can read here. Okay, provide blood supply and nutrient supply is the key one, although there are some other functions that we will talk about. Again, back to this image, we talked about the fibrous part. Now we're talking about this next layer, which is the vascular part. As you can see in the illustration, lots and lots of blood supply. Because what we talked about in the previous section on the nervous system is that neurons are very energy hungry, okay? They're energy hogs. They require a lot of oxygen and glucose because all of those neurons, there's lots of rods and cones in the next layer, and they're steadily consuming ATP, like burning through ATP like crazy. Just looking out at the room, you're burning ATP like crazy. So that means we have to have a steady supply of blood to keep the retina doing what it's doing. Okay, so it's a highly vascular layer. Vascular layer, as it approaches the anterior portion, the front of the eye, we have the iris, the ciliary body, and the choroid is really the vascular portion. So let's talk about the iris and the pupil, okay? The iris is a circular arrangement that regulates the diameter of the pupil, okay? The pupil is the hole, the iris is the donut-shaped muscle around it. And so regulating the amount of light is what this does, okay? If you go into a movie theater, all the lights are dim, your eye will dilate, okay? Dilation means to get bigger. And if you go out to the bright sunshine, let's say you're playing softball, baseball, and you have a pop flyer in the outfield, and you're looking up at the sun, okay? And the sun's blaring into your eyes, you're like, oh, I can't see anything. Your eye is gonna constrict, okay? Your iris will constrict. Okay, controlling that are two sets of muscles. You have the pupillary constrictor muscle, and you have the pupillary dilator muscle, which are controlling the diameter of the iris. If you look at this and you remember the mouth, remember the orbicularis oris, and then all of the radiating muscles that are running away from the mouth, it's kind of a similar orientation. This innermost here would be like the orbicularis oris, and here are those fibers that are kind of radiating away. Okay, one is to close, and one is to actually pull open. Okay. Pupillary dilation and constriction is a clinically available way to check for cranial nerve function. Okay, so we can check on cranial nerve 3 by just shining a little pen light into somebody's eye to see if we get bilateral or unilateral constriction based on that pen light. So if you see doctors going in, they're just doing a very quick cranial nerve check to see whether there's been any damage. If you were in a car accident, okay, if there's some, you're an athlete and you got knocked really hard and maybe damaged something, they're just gonna do a quick little check to see are all the cranial nerves working, and cranial nerve 3 you get checked by shine in the light. All right, so we talked about the fibrous layer. Whoops, sorry. Fibrous layer is the sclera and the cornea. The next layer is the choroid, or the vascular layer. The innermost layer is called the neural layer because it contains neurons. Okay? Special types of neurons, the rods and the cones, are the receptor neurons there. And we're going to get into more detail about the rods and the cones. You might have heard, maybe you had a science class or some other A&P class, cones tell you color and rods just tell you kind of black and white. It's really any photon of light would be detected by rods. Cones are really the high acuity, high definition vision, as well as specific wavelengths. Okay, so roughly red, green, blue, and then we can interpret things like yellow, orange, and purple. So here again, we're looking at this innermost layer. That's the retina, that's the neural layer. And there's a layer of cells here that is very highly organized. Okay, if you zoom in on that histologically, like from a microscope, you have a section of this. It's a very neat and orderly layer. Okay, the rods and cones are. And at the very back, we haven't mentioned this yet, but we have this kind of opening here where all of this nervous tissue converges and then exits out the back of the eyeball, bringing that data back to the brain via cranial nerve 2. Okay, 1 is for smell, 2 is for vision. Okay, so it's the cranial nerve 2. OOO2 touch and feel, so optic nerve. Okay, optic nerve. Also, that's where the blood supply enters posteriorly, is through the optic disc. Okay, it's where all those neurons are converging, also the blood supply. So this is an interesting thing, okay? If you think about the path of light, okay? Remember I said light starts with a light source, usually the sun or lights, a light bulb, okay? It bounces off an object, then enters into our eyeball. So here's the light entering through, okay? So here's the cornea, here's the iris, there's the pupil, there's the lens. The lens is focusing the light so it hits onto the retina at just the right depth, okay? So light is entering this way, okay, zoom into the big picture, this way. What's interesting is that the light photons have to pass through all of these layers of cells before they actually hit the tips here of the rods and cones. This section right here is the light sensitive portion. All these other cell bodies and parts of the cells are kind of in the way, right? So it seems like we would have really crappy vision. Okay, because if the light has to go through all that garbage, wouldn't we have really poor vision? And the answer is no. We actually have fantastic vision. Photons of light are so small that they kind of zip through unimpeded. We don't exactly know why. If you look at, this is a kind of a side note, but if you look at a squid or an octopus, the arrangement of the retina is reversed. It's the most bizarre thing in zoology. If you study, the visual systems in different animals compared to humans, it's reversed. So all of the light-sensitive cells are right up close here on the surface, and all the wiring is in the back for a squid or an octopus. But in humans, it's reversed. All the light-sensitive stuff is in the back, and all the wiring is in the front. And so people that are critical of a creationist viewpoint about the origins of human life would say, well, look at the eyeball. It is reverse engineered. If you think about the engineering principles, it doesn't make any sense why all of these cells would be in front of these light-sensitive portion of the rods and cones. What kind of a good God would design the eyeball like that? And it turns out that our vision is some of the best vision in the animal kingdom. Okay, so it doesn't actually play out that way because our vision happens to be quite good. Okay, in comparison to any other animal, we have some of the best vision as far as high acuity, see long distances, see short distances, color, black and white, all that stuff. So the answer is, I don't know. I guess I'll have to have that conversation with God at a later time. So, into the retina, okay? So the first layer Okay, if we're thinking about the first layer is the deepest layer, and that is the light-sensitive layer. Those are the rods and the cones because of their shape. These are kind of cone-shaped, these are rod-shaped, and they're embedded into this pigmented layer. Okay, the pigmented layer here is full of melanin. What does melanin do again? Sun hits the skin, melanin absorbs light, right? Protects us from UV damage. Melanin absorbs light. So we don't get scatter here. We don't have the light coming in and then it's kind of bouncing around. We actually come in, it's going to hit the rod and the cone, or it's going to get absorbed right away. That really helps us to have increased visual acuity, that is high specificity, high definition. In other animals that have more nighttime hunting and food gathering, so most mammals that are like squirrels and cats and dogs and deer, etc., you have the tapetum lucidum. Okay, the tapetum lucidum is that light reflective layer that would be right here that reflects the light back. It's kind of an iridescent yellow-green iridescent color. And when you shine a light out in the woods and you see the glow of the lights, that's the tapetum lucidum. Okay, most mammals have that. And it provides an additional sensitivity during nighttime, okay, low light conditions. Humans don't have a tapetum lucidum, okay? So animals have pretty good vision, but because the light is scattered, you have high sensitivity, but not high definition. So there's a trade-off there. And so just a couple points there. This is the pigmented layer into which the rods and cones are kind of embedded. Okay. The next layer is called the layer of the bipolar cells. Okay, it's one of the few places around the body. We talked in the nose, there's bipolar cells that are part of the olfactory receptor cells. These are bipolar neurons. They connect the rods and cones to the ganglion cells. Okay, now don't be confused. The definition of a ganglion in the nervous system is a collection of cell bodies in the peripheral nervous system. Okay, but these are called ganglion cells. Anatomists are just really screwy with names, and sometimes you're like, why did you name it that way? Because it's so confusing, but whatever. Ganglion cells, 1, 2, 3 layers. And at each of those synapses, between layer 1 and 2, there's a set of kind of these horizontal cells, okay? There's horizontal cells here, it looks kind of like an octopus. And again, between layer 2 and 3, we have another set of horizontal cells called amacrine cells. And that's another ball of wax. It's kind of a complicated way in which vision is managed with on-center, off-center sensitivity. So we don't have time, but all to say is these kind of horizontal cells manage those synapses in a way to highly specify regions of light and dark. Okay, so they're helper cells. They help manage how these cells are activated, and they can regulate those synapses appropriately. That's all we're going to say. What you should know is the rods and cones, the photoreceptors, the bipolar cells, and the ganglion cells. Ganglion cells have the super duper long axons that go back into the optic nerve. Okay, these have really, really long axons. So this is a histological sample. You can see the layers here. These are the rods and cones. Here's the pigmented layer, all that melanin. Here's the choroid, so vascular layer, lots of blood supply. Pigmented layer here, all these kind of stringy structures here, those are the rods and cones. These are the cell bodies of your photoreceptors, the rods and cones. And then here are the cell bodies of your bipolar cells, connecting rods and cones to the ganglion cells. And loosely here, just scattered throughout a little bit, you can see the ganglion cells. Okay, so here's your 3 layers, 1, 2, 3. Highly organized. When you look at it under the microscope, it's really beautiful to look at because it's so even and organized. This is what it would look like. This is the artist's rendition of that. Okay, 1, 2, 3 layers. All of those ganglion cells have an axon that's going to run towards the optic disc. Okay, that's the disc at the very back of the retina. okay, where there are no rods and cones because all those axons need to exit the eyeball, okay, as well as the blood supply. This is the optic nerve, cranial nerve, okay, cranial nerve 2. So this is where your blind spot would be, okay? The optic disc is where there's only axons and blood supply and no rods and cones, okay? So each eye has a blind spot. But the good thing about that is the position of the blind spot is medial, and the other eyeball tends to cover for the other eyeball, okay? The right covers for the left because the blind spots are in different locations. If the blind spots were both on the same side, then we'd have a problem, but they're not. All right, this is a histological section. It's from I teach another course called histology. I teach that in the fall, so if you want to have another course with me, need a biology elective, take histology. It's the study of human tissues. This is a sagittal section through the human eye. This is the eye. Here's the lens. Part of the lens here has been kind of scraped out due to processing. This should all be dark pink. This is the anterior chamber. Here's the cornea. This is the sclera. Here's the optic nerve. If you're going to zoom in here at the optic disc, we can see this here. Okay. All of these are axons. Axons are carrying that visual data back towards the brain. And here's that highly organized retina. Okay, the retina with the 3 layers of cells. 1, 2, 3. You can count them. Really, really cool that you can visualize all this organization histologically, microscopically when you look at it. It's awesome. It's beautiful. Okay? If you were to look, your doctor looks into your eye to do a fundoscopy. Okay? You have a fundoscope, the little fancy scope with the light on the end, and he or she is looking around. I don't know if I think I shared this last time, but my eye doctor in Little Rock, I needed to get some, I'm over 40 now, and so my vision is going. I had LASIK. Anyways, I was like, well, I gotta get contact lenses again, or just some glasses, because I just can't see at distance anymore. Set up an appointment at this random place I didn't know, so took a recommendation of a friend, and got into the examination chair talking with my doctor, young female doctor. I said, oh, I'm teaching at OBU. She's like, oh, I graduated from OBU. I'm like, that's, so it was really a kind of a cool thing, Dr. Wooten. Anyways, so she had a fundoscope and she was looking around my eye, just doing an eye examination. Here's the optic disc. You can see all the blood vessels are going towards, you would not be able to see the axons because they're much too small, but you do see the blood vessels that are servicing the vascular layer, or the choroid layer. So that's the optic disc. That's where your blind spot would be. Okay, it's slightly off center here just because the picture was taken on an angle, but this right here is a special location called the fovea. Okay, the fovea. The fovea is important. The fovea is the dead center of your eye axis. Okay, if I'm looking straight at you and you would put shine a light straight into my eye, you would be hitting the fovea. Okay, if you're reading a sentence and you're seeing the letters on the page, the part of your retina that is where the letters and the ink are hitting your eye is the fovea. Why is the fovea so important? Well, 1, it's in the dead center, and 2, it has an extremely high concentration of cones. All of this space of the retina, mostly rods. Right here in the middle of the fovea centralis is mostly cones. Talked about the different types of cells. If you do kind of a trick here, you cover one eye and you go in and out, you can see the plus sign will disappear. You can find out at which distance you can identify your blind spot. You can do this on your own if you have your laptop open. You need kind of a big screen. The phone might not work, but kind of cover one eye, look at the dot, and go close or far away until the plus sign disappears. Okay, left eye, looking at the dot, do right eye, you're gonna look at the plus sign. You can see where your blind spot exists. We talked a little bit about the chambers, right? The eye is a hollow organ. It's filled with two types of fluids, the vitreous humor and the aqueous humor. Okay, aqueous humor, aqueous like agua, meaning water, so it's a watery fluid. Okay, it's in the anterior chamber. Okay, and it diffuses through the walls of the anterior chamber into the scleral venous sinus, which has a unique name called the canal of Schlemm. Okay, canal of Schlemm. Fun word, fun name to say. And so aqueous humor is continually cycling through the anterior chamber, okay, and keeping that crystal clear so that light is entering from the cornea into the lens. That intraocular pressure inside all of the eyeball, okay, so it takes into account both the vitreous and the aqueous humor. There's a pressure that is always a little bit positive, okay, just slightly positive pressure, and that pressure can vary, and if it increases sometimes, you can get something called glaucoma. Okay, glaucoma occurs when you have an increasing in interocular pressure. That fluid is too much pressure. You're producing more fluid than it's draining. So that by itself is not so bad. It's just when that pressure starts to damage the optic nerve, okay, and you start to lose vision. Okay, so glaucoma is a disease where you have high interocular pressure that starts to press so much that it starts to damage the nerves. Of course, we could make the analogy to a camera. The eye has features that are similar to a very expensive camera. Okay? Back here is the light detector, right? Where the film would be in the olden days if you have a film camera, but now we have a CMOS sensor. Okay, that would be analogous to the retina. Okay, it's collecting the photons of light in a very particular pattern. Then we have all these lenses here, which are focusing and manipulating the light so that it hits the camera with just the right spacing and speed and all that good stuff. Okay. And we can have an aperture. The aperture controls the opening of the lens. Okay, that's like the iris. Okay, so you have lenses. We have the aperture. So a lot of kind of analogies to a camera. If anybody's into photography, here is the cornea, and here is the lens. Now, one thing that you need to know is when we talk about vision and how light bends, right? So as light is entering into the eye, it has to hit the retina at just the right depth. If it's too far or too close, then the picture will not be clear. Okay, so that it needs to converge at just the right spot. And so the lens will change shape to adjust for the right focal length and the focal depth. But the important point I want to say is that although the lens is the movable part that changes focal length, it's not the only thing that is bending light. The cornea actually bends light as well. It's this thick structure right here, and when light is entering in, it actually bends a little bit, okay? So the cornea bends light a little bit, and it's uniform. It never changes, okay? The cornea never changes unless you scuff it up or you cut it, okay? The lens does change, so this is the variable part. The iris here can control the amount of light that is entering into the eyeball. Again, if we are outside and we have all that light flooding in, it's just going to overload all those rods and cones, and they're going to be completely blasted and going to have blindness, right? So when you're outside, bright sunny day, your iris is constricted. All right. The lens and the lens fibers, there are an arrangement of collagen fibers inside the lens that are flexible, elastic, and optically clear that allow for light to bend in just the right way. Okay, a cataracts is a condition, a disease where the lens has lost its transparency. There's parts of the lens which are occluded, and it can impede the path of the light going through the lens. Okay, so a cataract occurs in the lens and it blocks the light. Okay, it's not uniform in its clarity. Okay, so a little bit of physics. If you've taken a physics course at OBU, or maybe you had physics course in high school, you learned about some of the principles of light. Okay, when light goes through the lens, it inverts, right? Okay, right to left and top to bottom, and we can change the path of light by changing the curvature of the lens. Okay, we can control focal point and focal distance. Okay, so if we know that there's a very specific distance that we need to focus something on, if we have a very far distant object, or if we have a very close object, we need to change the lens to adjust for the focal length. Okay? And by changing the thickness or that curvature, we can make the proper adjustment to make sure that we have the proper focal length. Okay? Accommodation. Okay, that's a vocabulary word you need to know. Accommodation is the changing of the thickness of the lens. Okay? It's how the lens is fat or thin to accommodate far sight or near sight. Okay? A far object or a near object. Okay? Astigmatism is a condition or a disease state where the quality or the curvature of the lens is not uniform. Okay? And so there's an amorphous shape to the lens, which allows for light to kind of diffract a little bit, okay, and not in the right way. And so astigmatism can be an indication of poor vision. Okay, you can have bad vision because your eyeball is a little bit elongated or a little bit shorter, but it's complicated by astigmatism, which is a disorderly scattering of the light because your lens is not quite purely shaped in a disc. Okay, so here are the two ways. You can see just a subtle change in the thickness. It's a little bit thicker here and a little bit thinner here. Okay, the thickness here would allow for a close object, like imagine you're taking a sliver out of your finger, okay, and that light is going to be perfectly focused onto the retina, not too deep and not too shallow. Whereas if you're focusing on a far object, like a traffic light or a mountain that's far away, okay, it's going to be kind of thin to appropriately bend the light so it again focuses on the retina at the proper focal length. Visual acuity is usually measured in 20/20, although there's diopters as well. You might see like a minus number if you're nearsighted. My vision back in the day was like minus 5.75 and minus 4.75. It was really bad. But -1, -3, those are common diopter numbers. Okay, important point. Any light that passes through a lens, we learned this in physics class, inverts the image. Top becomes bottom, okay, and left becomes right. Okay, so here's the picket fence. The pink is on the right, the blue is on the left. When it hits the retina, everything is inverted. Okay, and magically, not magically, but by design, your brain figures it all out. It re-inverts it. Okay, so the way that a fancy camera controls for different focal lengths is to move the lens in and out, okay? So we don't move the lens, we thicken the lens. Okay? And if we need corrective lenses, let's say our eye shape is not perfect and we need corrective lenses, either contact lenses or reading glasses, well then we can add an additional layer of bending the light that helps to focus. Okay? Camera moves the lens, eyeball thickens the lens, the curvature. Okay? We call myopia nearsightedness. It just means that the light is focusing too shallow. And hyperopia is farsightedness. It just means that your eyeball is a little bit too short for the axis and it's focused too far. So the type of lens that is required to correct that will be slightly different shape. You can also have surgical correction. There's something called LASIK. If you didn't know what that stands for, it is the laser-assisted in situ keratomyoleusis. Okay. There's also PRK. Okay, PRK. There's two methods. They're both doing the same thing, which is essentially reshaping the surface of the cornea. The LASIK uses a flap, and the PRK does not. That's really the only difference, okay? LASIK, you make this flap, and you kind of flap it open, and the laser goes dit, 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 and it resurfaces your cornea, and you put the flap back, and it heals. PRK is usually, if you don't have really thick corneas to begin with, you're not a good candidate for LASIK, but you might be a candidate for PRK. And they'll just do the laser correction right on the surface. The healing is a little bit different because there's no flap to return. I had LASIK. If you're squeamish about stuff kind of cutting your eyeball, I get it. I was there. It turned out to be one of the best decisions of my life because I had contact lenses for forever. I had reading glasses, and for a good chunk of my 20s and 30s, and some of my 40s, my vision was great. Now as I'm aging, I need contact lenses again, which I'm bummed about, but that's life. All right, let's zoom in on some details here in the retina. Let's talk a little bit more about rods and cones and the mechanism by which these cells harvest light information. Okay, that's what vision is about, right? What are we doing in the eyeball? We're collecting light information. Because as light is scattering around this room, the classroom, your bedroom, your dorm room, the library, outside, whatever, there's lots of information. And all of that information about the external environment is hitting the retina, and you're collecting it, and it's in immediate responsiveness. You can distinguish where you are and moving objects, etc. You can read. So we have visual pigments that are in the rods and cones. Okay? They start with something called vitamin A. You can get vitamin A derived from beta carotene. Eat your orange vegetables, your carrots and your squash and your whatever, okay? Mango, anything that's orange or yellow has vitamin A in it, okay? So eat your carrots. It will give you retinol, okay, OL, which can be converted into retinal. Okay? And in chemistry, you'll learn the difference between an alcohol and an aldehyde. Okay? An alcohol has a single bond to an oxygen and a hydrogen, and an aldehyde has a double bond. Those can be converted back and forth, no problem. One is in retinol, one is a retinal. Okay, I'm kind of making that really obvious. Retinal gets converted into retinoic acid. Okay? That gets oxidized and it's kicked out. Okay, but we need lots and lots of this particular pigment because of this reaction that occurs. Okay, so there's a protein, and then this cofactor is inside the protein, and when light hits it, it changes shape. That's super cool. That's how vision works. Let's look at that. Okay, here's a pigmented layer. Here's a rod. Here's a cone. This is called the outer segment. It's where the tips here look either rod-shaped or cone-shaped. That's why they got their name. The outer segment of these photoreceptors have layers and layers and layers of plasma membrane. Now, some of them are inside, some of them are outside, but they're still, if you think about a plasma membrane, it's basically lipids and there's proteins embedded in there, okay? Lots and lots of layers. And that's where these photopigments are, the rhodopsin. Okay, and then we have the rest of the cell, the nucleus, and if this part of the rod or cone is getting excited, the whole cell will become depolarized or hyperpolarized, in fact, and then communicate to the bipolar cell, and it'll start the chain of action potential sending to the brain. Now, what I said before is that the fovea centralis, if this is the eyeball, okay, here's the lens, here's the cornea, and the light is entering this way, here's all of the retina. So when we look at something, we have the center of our vision, that's where we like read letters on a page, and that's where we direct, the center of our vision is the best quality vision is always in the middle. But if I'm looking at you, there's stuff over here, that's my peripheral vision. I still have visual data, it's just not in the center, but it's still helpful to me. Like if something's coming at me, I could dodge it because I see something out of my peripheral vision. This is your peripheral vision here, okay? This is the center of your vision, that's the fovea. There's a little dimple right there. Okay, and the dimple actually is bending some of that wiring out of the way so that there's direct access of the photons of light to the rods and cones. So he's talking about that wiring problem, the wiring's in the way. At the fovea centralis, it's those rods and the bipolar cells and the ganglion cells are kind of bent out of the way. So it's literally not a problem. Anyways, this graph shows the distribution of rods and cones proximate to the fovea. Okay, let me explain. So I said most of the retina is composed of rods, okay, shown in blue. Okay, so here's the surface of the retina. If you take this as the retina here, you're going to flatten it out and lay it down like so. Okay, here would be the surface of your retina. The height here would represent how many rods or cones. And in blue, you can see most of the space is all rods, 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 rods. Rods drop off, sudden spike with cones shown in red, right? This spike right here, that is right at the fovea centralis, okay? You can see that drops off with rods and it spikes with cones. Cones are high definition and color vision. Now, there are some cones sprinkled throughout, okay? So we do have color vision everywhere. It's not like we don't have color vision in the periphery, but we just have more rods than cones. So this map here shows a distribution of the rods and cones. It's really a kind of a sweet map. Guess what that is? That's the blind spot. That's where your optic disc are, because you have no rods and cones there. Okay, that would be right where this guy is. All right, now, remember this location right here, these layers and layers in the discs, okay? These are discs of plasma membrane, essentially. If we zoom in on that part, we would see all these layers and there's proteins that are embedded, okay? So here's the plasma membrane. Here's a protein that's embedded into these plasma membranes. So here's these discs, okay? This is a protein called rhodopsin. Rhodopsin has retinal embedded in it. Okay, so it's a protein with a molecule inside of it. When this molecule of retinal receives a photon of light, it changes shape. Okay? There's a chemical reaction that is triggered by a photon of light hitting retinal. Okay? And that will cause rhodopsin to change its properties. So the way that this protein receives that light depends on what type of protein it is. Okay? And different types of cones can receive red wavelength or green wavelength or blue wavelength better or worse. Okay? If we have color blindness, it means that we have an inability to detect certain colors, meaning some of those proteins are absent or defective. Okay, so if we have blue cones, they're not colored blue, but they are sensitive to blue wavelength of light. They have an optimal window of wavelength that they detect. And we have ones for green, we also have ones for red. Okay, and between those three colors, our brains can interpret yellow, orange, and purple. Okay, and in black here, those are the rods. Those are kind of in the middle of the whole wavelength. They have an optimal length kind of around greenish. And so that's the optimal wavelength for detecting ambient light. So each of the rods and cones have an optimal wavelength which they detect, and by that mechanism, your brains can interpret different colored objects. Okay, so if you are colorblind, you might be red-green colorblind, and you would not be able to distinguish the red here should have the number 12 showing up, and here you should be able to see 74 or 71. I don't know. Looks like 74 or 71. I think it's 4. All right. So how does that work? Okay, talking about photons of light. Photons of light comes from a light source, the sun or a light bulb, okay, or a flame. Okay, the photon bounces off the object, enters into the eye, strikes retinal. Okay, that's the photopigment that is sensitive to light, and then it changes the shape. Okay? It changes from 11 cis retinal to 11 trans retinal. Let's look at how that would work. So each of these proteins that have retinal in them cycles because they're going through cycles of being stimulated and then being reset and then re-stimulated. Okay? So here we have opsin. We have 11 cis that's inserted into the opsin, becoming rhodopsin. Okay, rhodopsin has the 11 cis inside of it. A photon of light hits it, goes from a bent conformation to a straight conformation. Okay, it goes from a cis conformation to a trans. Okay, that shift causes the 11 trans retinol to be ejected, and a momentary shift in the protein properties, which will trigger a response of the cell. Okay, and it requires an enzyme hydrolyzing ATP to reset opsin into rhodopsin. Okay, so as I said, neurons and photoreceptors are very energy hungry. How do we get that ATP? Well, we got to burn glucose and oxygen, got to metabolize them, right? Okay, so all of these things are happening in the distal portion of those rods and cones. All right, so those are some of the details about the anatomy of the eye, the accessory structures, the eyeball itself, the layers, and a little bit of kind of molecular biology, frankly, with how rhodopsin receives light data. Now we're going to talk a little bit about the visual pathway, okay? I'm just about out of time for this lecture, but I'm going to briefly go into visual pathway. When I say visual pathway, I want you to think about the visual field. Your visual field is whatever's in front of you at any given time. If you're sitting in your car, your visual field would include your windshield, your steering wheel, all the controls, the road, okay? Whatever you can see when your head is still, that's your visual field. Okay? You have your right visual field and your left visual field, okay? That is the space to your right and the space to your left. Both your right eye and your left eye are picking up your right and your left visual field. Of course, it's inverted, okay, right to left and top to bottom. So as that light enters into the eye, hits the retina gets excited, converts that data into an action potential. It leaves the optic nerve, travels through the optic chiasm, enters into the thalamus, okay, and the lateral geniculate nucleus, okay, the LGN, is the particular nucleus that receives visual data, synapses there, and then another neuron projects all the way back to the optic, the primary visual cortex, which is the occipital lobe. Okay, so this is the pathway. Okay, what is the sequence of events that bring information about your visual field into the eye, activates the retina, converts it into action potential, travels along the neurons into the thalamus, and then finally brings it to the primary visual cortex. That's the pathway. It starts with those photoreceptors, the rods and cones that we saw, and it ends at the visual cortex, okay, occipital lobe. Okay, there's this information about on-center neurons and off-center neurons. Don't worry about it, skip over it. I'm not going to ask you about on-center, off-center. It is much more detailed understanding about how rods and cones behave with respect to each other to really refine visual field. I'm not going to test you on it. Don't worry about on-center, off-center. All right. So there's lots of things that your eye does with visual data, okay? And I'm going to go through them on this slide in the next lecture, okay? See you guys in class.